Ok, bonito jueves a todos. El día de hoy tenemos una sesión que está a cargo de la doctora Sandra Teniente, es nutrióloga con maestría y doctorado. Nos va a platicar acerca de un proyecto de investigación que está en vías de ser aceptado, esperemos, Sandra, que así sea, bueno, ahorita nos va a platicar ya acerca de qué se trata. La doctora Sandra Teniente se encuentra ahorita en el Hospital Universitario de Saltillo, de la Universidad Autónoma de Coahuila. Les recuerdo que estamos grabando este programa. Cualquier asunto que sea confidencial, obviamente nombres de pacientes o de personas y de proyectos, cualquier información sensible no debe de ser tratada por este medio. Al término de la presentación de la doctora Sandra Teniente, tendremos la oportunidad de hacerle preguntas y hurgarle el cerebro para poder extraerle la mayor cantidad de información que sea posible. Agradezco la presencia bueno, todo mi equipo de laboratorio, la doctora Gaby y el doctor Lauro. ¿Quieres iniciar, Sandra?
Sí,
El
claro,
micrófono
muchas
es tuyo. Si
gracias.
tienen preguntas, lo normal es que pueden interrumpir conforme van eh, surgiendo las preguntas para poder sacarle más provecho y no perdernos que de repente se van acumulando las preguntas y no sabemos qué hacer. Pero entonces ya saben, la dinámica es abierta. Adelante, Sandra. Gracias por estar con nosotros.
Gracias.
Y disculpen, esta es la primera vez que vamos a empezar a presentar los webinars en inglés. Entonces, nuevamente fue una conclusión a la que llegamos Gaby y yo, porque nuevamente los webinars los subimos a Facebook, a YouTube, y más de la mitad de los que nos visitan en esos dos canales no son de origen latinoamericano, no manejan el español. Entonces, para poder abrir un poquito más la capacidad de proyectarnos como grupo de trabajo, ¿va? Ok. Adelante, Sandra.
Okay, first, I'd like to thank you for this kind invitation. It's truly a pleasure to join you today and share some of my work. I have always thought that these type of webinars are wonderful opportunities for exchanging knowledge, discussing new findings, and well, learning together as a community. For those who don't know me, I'm Dr. Sandra Teniente. Currently, I lead the nutrition and cookie department, as you can see. And I also collaborate with the team of CIGEN, the Center of Genetics and Genomics Investigation, working closely with Dr. Gaby Ortiz, Dr. Rafael, and Dr. Sunday on several research projects. Today, I will share with you the findings of a research study I conducted at Universidad Autónoma de Coahuila, which led to the writing of a results article currently under publication in the journal Current Nanoscience. In this study, we synthesize gold nanoparticles using a green reduction technique, employing polyphenols from pomegranate peel and coffee pulp as reducting and stabilizing agents. Then we evaluate their antiproliferative effects on different cancer cells. In this project, our primary goal is to contribute to the development of innovative alternatives for therapy and cancer treatment. And well, before diving into our methodology and key findings, I'd like to take a moment to outline the current challenges in oncology that motivate this work. As a brief introduction, cancer is a group of diseases characterized by the development of abnormal cells that divide and spread uncontrollably in any part of the body. This pathology is one of the leading causes of morbidity and mortality worldwide. According 2020 GloboCan report, an estimate of 19.3 million new cases, cancer cases, and around 10 million deaths associated with this disease were recorded. There are different therapies for the treatment of this disease. The most commonly used are chemotherapy and radiotherapy. However, the side effects that they cause in patients are severe, deteriorating their overall health and weakening the immune system. For this reason, alternative therapies have been proposed, such as the use of polyphenolic compounds. Well, polyphenols are chemical compounds found in plant-based foods, such as fruits and vegetables, and they have attracted the attention of researchers and physicians in recent decades due to their multiple biological activities. In Mexico, we have a vast diversity of food with high polyphenol content. Notably, we have pomegranates and coffee. Pomegranates are cultivated in regions such as Baja California, Sonora, Zacatecas, Tlaxcala, and others, while coffee is characteristic of states like Chiapas, Oaxaca, Veracruz, Guerrero, and San Luis Potosí. However, both pomegranate peels and coffee pulp are underutilized byproducts that, despite their abundant polyphenolic compounds, generate tons of waste every day. We have analyzed the polyphenols present in pomegranate peel and in coffee pulp, identifying promising antiproliferative effects across various cancer cell lines. These findings suggest their potential as therapeutic agents for oncologic treatment. However, one of the challenges we have faced is that polyphenols are very sensitive compounds that degrade under adverse environmental conditions, including the exposure to light, particularly ultraviolet light, and also to oxygen and high temperatures. So to address this challenge, we have explored the use of nanotechnology, specifically by incorporating gold nanoparticles. So this strategy enhances polyphenol stability and efficiency against these degrading factors. Well, next I will discuss the methodology used for polyphenol extraction, synthesis of gold nanoparticles, and finally, the analysis of the treatment effects on cell viability across different cell lines. Well, our first step involved obtaining pomegranates from a local market in Saltillo, Coahuila. The peels were removed, washed, and dried at 50 Celsius degree for 48 hours. Similarly, coffee pulp was sourced from a coffee plantation in La Huasteca Potosina and dried under the same conditions. Following this, the materials underwent grinding and sieving to achieve particles of approximately 100 microns in size. Next, polyphenolic compounds were extracted using ethanol and a hybrid ultrasound microwave method under the parameters that are displayed on screen. This hybrid method had demonstrated high extraction efficiency, reduced extraction time, and lower use of solvents. Subsequently, the extracts were filtered and subjected to column chromatography, using as a stationary phase amber light. Amber light is a resin that enables highly efficient polyphenol purification. And as a mobile phase, we use water and ethanol. The ethanolic phase was recovered and dried at 50 Celsius degrees for 24 hours to isolate the polyphenols. This process allowed for effective separation and purification of the target compounds while minimizing degradation of thermal sensitive compounds. Once the polyphenolic compounds were recovered, they were identified using high-performance liquid chromatography coupled with mass spectrometry. And the results indicate that among polyphenols derived from pomegranate peel, we identify punicalagin, pedunculagin, and ellagic acids. These findings align with those reported by Matthew and collaborators on 2021, who analyzed polyphenols in pomegranate peel from the wonderful variety, and they detected the primarily compound was from elagitanine family. Additionally, we identify alpha punicalagin and beta punicalagin as the predominant compounds corresponding to the peaks 2 and 3 in our chromatogram. In contrast, the polyphenols derived from coffee pulp were denominated by members of the hydroxycinnamic acids, tyrosols, dihydroflavonols, elagitanins, alkylphenols, and lignans. These findings align with Vega y colaboradores in 2018, who reported that hydroxycinnamic acids and their esters are the most abundant polyphenolic compounds in Arabic coffee pulp. And the most abundant compound identified was chlorogenic acids, as you can see in the peak 2 of the chromatogram. And these results are consistent also with Vega y colaboradores, who confirm chlorogenic acid was a predominant phenolic compounds in Arabica coffee pulp. We now present our optimized protocol for gold nanoparticle synthesis.
I'm sorry, Dr. Teniente.
Yes.
Are those chromatographic profiles similar? If you look at coffee brew, you know, like coffee brew, not just the pulp, and if you look at the pomegranate pulp and not just the whatchamacallit?
Peel or husk.
The husk, yeah. Do they have different, I suppose they have different contents of all these substances? You wouldn't expect the same profile?
They have different compounds. In pomegranate husk, we find also alpha and beta punicalagin, but we also can see another compounds. And for the coffee pulp, the chlorogenic acid is the most abundant compound, but in the coffee we have acid. I don't remember the name of the compound, but it's a
different profiles.
Yeah,
different chromatographic profiles.
Yes.
So the benefits, the antiproliferative benefits of coffee pulp and pomegranate are not going to be exploited in a normal coffee brew like I'm drinking right now?
No.
Or they might be. No, they're not present. Okay. I was thinking of drinking more coffee.
More coffee, yes.
Okay.
Obviously, coffee have a lot of polyphenols, so you will have a good amount of polyphenol compounds. But
not as much as the pulp.
In the pulp, we use like pulp and husk because they are like waste products. So a coffee pulp is usually only waste because you can use it for compost or for animal food because they have like a high amount of caffeine. So it's not utilized and it's only waste.
Okay.
Dr. Sandra, is it better to drink cold coffee because of, the degradation? And just curiosity.
Yes, a lot of polyphenols lose their properties at 80 degrees. So if you use like boiled water, probably you're going to degrade a lot of the polyphenols. So maybe like temperature below 70 degrees or less will be the optimal.
Well, good coffee makers know that you shouldn't use boiling water to prepare coffee. But
yes.
I mean, so is Gabby saying we need to drink iced coffee?
Iced coffee?
Iced coffee.
Yes.
But you still have to brew it with relatively boiling water. So, okay. So let's just drop coffee out of the question. We'll have to chew on coffee beans then. Thank you.
Coffee beans. Right. Another question? Okay, I will continue. Now I'm going to present the optimized protocol for the gold nanoparticle synthesis using, as I say already, pomegranate peel and coffee pulp polyphenols as reducing and stabilizing agents. Well, following the isolation and characterization of polyphenols from the both byproducts, we proceed with the synthesis of functionalized gold nanoparticles using a green reduction approach. The protocol involved reacting 1 millimolar chloroauric acid solution with polyphenol extracts from pomegranate peel, coffee pulp, and a mixture 101 of both. We use it as both reducing and stabilizing agents. Traditionally, reducing agents use are sodium borohydride and hydrazine. They are the most often used, but green synthesis employ eco-friendly alternatives, like in this study, because polyphenols act as reducing agents by donating electrons to chloroauric acid, promoting the reduction of the salt and forming gold nanoparticles, while also acting as stabilizing agents, preventing that the nanoparticles suffer some kind of aggregation. Then the solutions underwent to continuous agitation for 24 hours, after which a distinct color change to kind of reddish was observed. This chromatic transition is characteristic of gold nanoparticles that have a diameter of 20 nanometers or less. This was reported for Chang and collaborators in 2019. Subsequently, 1 milliliter aliquots from each solution were collected for concentration quantification through lyophilization and gravimetrical analysis. The concentrations, are those displayed in the screen. And then we, from this stock solution, prepare 3 serial dilution, 110, 100, and 1000. This was for evaluate dose-dependent effects. For the nanoparticle characterization, we employ 4 analytic techniques. X-ray diffraction. This was used to determine crystallography structure. Dynamic light scattering for the hydrodynamic size distribution analysis. Fourier transform infrared spectroscopy to identify the surface of the functional groups, and scanning electron microscopy for the morphological evaluation of the nanoparticles. First, we have the results of the X-ray diffraction. Figure 4 displays XRD patterns of the synthesized nanoparticles from pomegranate peel, coffee pulp, and this is from the mixture of both. And they are compared against the reference gold diffraction patterns. As you can see, all the samples exhibit characteristic face center cubic gold peaks, with the most intense at 111 reflection, and this was used for the crystallite size calculation. For this, we use the Scherer equation, and the sizes that we found are 21.98 nanometers for pomegranate peel nanoparticles, 19.49 for coffee pulp nanoparticles, and 20.20 from the mixtures. This confirmed the uniformity of our green synthesis approach. Then the results for the dynamic light scattering analysis, that is a well-established technique for measuring particle size distribution, but in a colloidal suspension. The results from the gold nanoparticles from pomegranate peel reveal that our nanoparticles exhibit diameters below 100 nanometers, with a predominant size peak at 25.4 nanometers. This distribution indicates a highly sample homogeneity that is a critical quality parameter for biomedical applications. Then we have the results for the coffee pulp polyphenols, functionalized nanoparticles. And this analysis confirmed that 90% of the particles exhibit diameters below 100 nanometers, with a model size of 3.6 nanometers. And this narrow size distribution demonstrates that stabilizing efficiency of coffee pulp for gold nanoparticle synthesis. And last, nanoparticles synthesized with a hybrid of pomegranate husk and coffee pulp extract demonstrate diameters below the 100 nanometer diameters, with a predominant peaking at 29.6 nanometers. This consistency in core size across all three formulations highlights the reproducibility of green synthesis protocols. And now the results for the Fourier transform infrared spectroscopy. In figure 8, we can see the FTYR spectra of both the crude extract of pomegranate husk below and the functionalized gold nanoparticles on top. And we revealed two key features. First, a distinct absorption band at 1600s. This corresponds to the carbon and carbon double bond aromatic ring stretching vibration. This is consistent with report spectroscopy data for polyphenol compounds. And we also identify intense band at 3200, and this is characteristic of phenolic hydroxyl groups from the HDP units. This confirming successfully polyphenol functionalizations of the gold nanoparticle surface. And finally, on figure 9, we can observe the FTYR analysis of coffee pulp extract and the gold nanoparticles with the coffee pulp polyphenols. And we see a lot of bands corresponding to hydroxyl groups, alkanes, and carboxyl acids functional groups. These spectra signatures closely match the expected vibrations modes of chlorogenic acids and caffeic acids. That was the dominant polyphenol constituents of our extract. This spectroscopic evidence confirms successful functionalization of the gold nanoparticles with the coffee derived phytochemicals. And finally, the FTYR analysis of the hybrid, the mixture between polyphenols from pomegranate peel, coffee pulp, and we see two defined spectral signatures. One, well, the signal of a hydroxyl stretch at 3200, characteristic of the phenolic group abundant in both puricalagin and chlorogenic acids, and two assign at 1600s absorption band corresponding to the conjugate carbon-carbon vibration that is also seen in aromatic rings, consistent with the core structure of these polyphenols. Now we will focus on the effect of polyphenol functionalized gold nanoparticles in different cell lines. Well, 4 cell lines were reactivated for this study. HeLa, that is a cervical cancer cell line. A549, that is a lung cancer. MDA-MB is breast cancer. And finally, HIC293, that is a non-transformed embryonic cell line. And this is going to serve us as a control. All cell lines were exposed to 3 distinct treatments: gold nanoparticles with pomegranate pulp polyphenols, with coffee pulp polyphenols, and the hybrid, the mixture 101. And these treatments were exposed to 3 distinct concentrations: 1,110 micrograms per milliliter. Cells were seated at 9,000 cells per well in 96 well plates and allowed to adhere for 24 hours. Subsequently, the medium was replaced with the gold nanoparticles suspension that I previously described. Following the treatment, cells were incubated with MTT reagents for 4 hours, where the mitochondrial reductase activity in viable cells reduce the MTT to purple formation crystals. These were solubilized with DMSO, and the absorbance was measured at 590 nanometers. And all the experiments were conducted in triplicate with untreated cells as negative control and 1% return as positive cytotoxicity control. Well, the results show that in HeLa cell line, treatments 1 and 2, 4 and 5, 7 and 8 had an antiproliferative effects by reducing cell viability to less than 75%, which is significant according to the ISO 10993 standards. Moreover, when comparing this data with our negative control that consists in untreated cells, we observe a significant difference. We also noted that treatment 7, that is the highest concentration of gold nanoparticles with pomegranate, coffee pulp, sorry, showed the greatest reduction in viability. This can be explained because chlorogenic acids, as the main compound, a compound in the extracts of coffee pulp, can increase caspase expression, activating apoptotic pathways, while also increasing the expression of BAX, that is a pro-apoptotic protein, and decreasing the expression of BCL2, an anti-apoptotic protein suppressor. This was reported in this cell line, HeLa. In A549 lung cancer cell line, we also observed that the treatments 1 and 2, 4 and 5, and 7 and 8 exhibit antiproliferative effects lowering the viability in less than 75%. And we also notice that this effect is dose dependent. This can be explained primarily through the chlorogenic acid ability to activate the expression of caspase 3, 8, and 9 in this cell line. Additionally, punicalagin was found to upregulate caspase 3 and 9, while also increasing ROS generation, leading to a mitochondrial dysfunction and finally to apoptosis. This in A549 cell line. In the MDA-MB cell line, only treatment number 7, corresponding to the nanoparticles with coffee pulp polyphenol, show an antiproliferative effect. This may be due to the nature of this cell line, which is a highly aggressive and invasive cell line because it's a triple negative. This means that it lacks estrogen receptors, progesterone receptors, and HAR amplification, and these make it more resistant to apoptosis. Additionally, these cell lines carry some types of mutations in genes like TP53, which is highly involved in cell cycle progression. However, some studies have shown that chlorogenic acid functionalized nanoparticles can induce apoptosis in another breast cancer cell lines. This by inhibiting the PY3K AKT mTOR pathway, and this mechanism bypass conventional hormone receptor dependent apoptosis resistance, offering a promising therapeutic strategy for treatment of other cancer cell lines, especially MDA-MB.
Excuse me, Dr. Teniente, why would you use Triton X as a reference?
Sorry?
You're using Triton X, a detergent, as a reference?
This is like our positive control.
Okay.
To
it's because it just lyses all cells, right?
Yes, lyses all the cells. So we have like that control, like a positive control, and cells without any treatment as a negative control.
Okay.
And in the non-transformed HIC-293 cell line, none of the treatments show antiproliferative effects because neither reduce cell viability below 75%. This demonstrates the selective action of both polyphenols and gold nanoparticles against healthy and non-transformed cells, like is the case of HIC-293. Also, Perumal and collaborators in 2019 observed proliferative, anti-apoptotic, and antioxidant effects of standard nanoparticles on HIC-293 cell line, and also they detected a void in caspase activation. Furthermore, we assess the cell death using the annexing staining protocol. For this assay, we select the cell lines HeLa from cervical cancer and A549 pulmonary cancer, because we see that these cell lines have the highest response to the treatments. They were seated in microplates and exposed to the 3 treatments of nanoparticles. Nanoparticles with pomegranate peel, coffee pulp, and the mixtures. And they are exposed to only the higher concentration, that is 1,000 micrograms per milliliter. Subsequently, they were incubated for 24 hours at 37 degrees Celsius. Then they were washed with PBS, and the dual reagent was added, which contained an apoptosis detection reactive, a necrosis detection reactive, and finally a binding buffer. They were then incubated for an additional of 15 minutes at 37 degrees Celsius. Cells were covered and immediately imaged using a fluorescence confocal microscope equipped with a dual filter set for apoptosis and for necrosis detection. Simultaneously, imaging was required. For positive control samples, cells were pre-treated with an apoptosis inducer. In this case, we use staurosporin at a final concentration of 2 micromolar for 4 hours. The fluorescence microscopy images were analyzed using a software, the Cell Profiler software. This was used for a quantitative assessment. This is the software. Cell Profiler is an open source image analysis platform designed for quantitative evaluation of biological microscopy data. The software supports processing of images acquired through different microscopy modalities, and it gives you a precise identification of biological objects, in this case, well, cells and also organelles.
So and this is free software?
Sorry?
Cell Profiler's a free software?
Yes, it's free.
And it works on Mac and Linux and Windows?
I don't know. I use it in Windows, and I don't have any issue. It's great because it identifies cells, nucleus, or other organelles, and it gives you like numeric data.
Okay, that's very nice. I didn't know about the software. It looks nice. Is it user-friendly?
Yes, it's so easy to use.
Okay, thank you.
Here we can see the microscope fluorescence images of HeLa cells treated with gold nanoparticles, functionalized with polyphenols extract for coffee pulp. And we are in panel A. We can see the apoptosis specific filter, and we see a green fluorescence. Indicates the cells that are undergoing apoptosis, programmed cell death. And in panel B, we can see, I don't know if you see correctly, but we can see like a reddish stains. And this is under the necrosis filter. Identifies cells with a severe membrane damage or unprogrammed cell death. This dual filter approach enables clear differentiation between apoptosis and necrosis, providing.
So you're expecting
Yes.
your treatments to induce apoptosis, but not necrosis. Is that right?
We didn't know what type of death was occasioned the polyphenols and the gold nanoparticles. So we want to see if it was apoptosis or necrosis. In this case, we identified that the primarily type of death was apoptosis.
Okay.
Yes. Okay. Well, it's also important to note that when we observe both sides, if you see like the signs from apoptosis and necrosis in the same image, it is telling you that these reddish cells are not really in necrosis. They are like in a late state of apoptosis. Yes. Well, this is like the information that gives you the cell profiler program. See, we can, this is like from HeLa cells treated with the gold nanoparticles and pomegranate, coffee pulp extract. In the image in the A section, we can see the original image that you see earlier, but only in like a gray scale. In B, this is like a pseudo-color image that display individual cells. This is like individual cells that the program identify. And in, this is in C. In B, we can see like the nuclei of the cells. And in the D, we can see like the information. The number of objects that were identified, number of pixels, and the cover area. In these images, the cover area of cells that are in a state of apoptosis was 10.1%.
So does that mean that 10.1% of the cells are necrotic, or 10.1% of the field of view it has cells?
Yes, exactly. So in the microscope fluorescence, we only see images, but there's no way that you can identify how many of that cells. Yeah. So this program gives you some kind of information. In this case, like 10% of cover area of cells in a state of apoptosis, and we took that percentage to compare with other treatments.
Okay.
I have like a lot of image, but rather than displaying all the microscope image, which will be so impractical due to the large number and the time we have, I only will present the extract quantitative results. Sorry, I apologize for the slight language, but let me explain. This shows the comparison of the total cover area percentage from the fluorescence microscopy images under apoptosis and necrosis filter. We observe in this table HeLa cell line with those 3 different treatments. The first is the treatment with gold nanoparticles with pomegranate husk, then with the mixture, and finally with the coffee pulp extract. We can see that in the first treatment, 6.4 of the area total area of the cells are in an early state of apoptosis, and 4% are in the late state of apoptosis. Why do we know that these 4% are not in necrosis? Is because it is shown if it detects both necrosis and apoptosis, it means that they are in a late apoptotic states. If the cells were only necrosis, we only see the red sign and not the green. I don't know if I explained it right. Or anyone?
I just have a question. What does this mean? I mean, with the coffee pulp, the column on the right, you get relatively similar amounts of late and early apoptosis, whereas the pomegranate peel, I think, no, the coffee, the first one.
Sí, ese es pomegranate
pomegranate only. It's skewed. It seems to be the proportion of cells that are in an early apoptotic stage are more abundant than those in late stage. What does this mean? What do these differences tell us?
We believe that when we activate and cultivate the cells, they grow overnight. So there are cells that are born first or divide first than others. So they start the apoptotic process earlier. That's what we think that's because we have that difference. Like both are in apoptosis, only in different times of the cell cycle.
Okay, but these were all assessed at the same time? I don't know, 24, 48 hours after treatment, or how much time after?
We activate and they stay 24 hours, and then we proceed to
Read.
To read. Yes.
Okay. And did you see the same behavior with other cell lines other than HeLa?
HeLa, yes, I have these results and from A549 cell lines.
I'm sorry.
Yes, don't worry. And in the treatment with gold nanoparticles with the mixture, we can see that 8.5% are in early apoptosis and 5.2 in late apoptosis. And in the treatment with gold nanoparticles with coffee pulp, 10.1% was on early apoptosis and 8.9 on late apoptosis. We can see that the effect is most important in the treatment with the nanoparticles with pomegranate, with coffee pulp. And finally, we can see the comparison of the total cover area percentage from the fluorescent microscopy images under apoptosis and necrosis filter. And we observe that the line A549 with the pomegranate pill treatment, 6% are in early apoptosis and 1.8 in late apoptosis. And in the mixture, 5.6 are in early and 2% in late. And finally, with the coffee pulp nanoparticles, 7.9% are in early and 2.5 in late apoptosis. So as a general conclusion, gold nanoparticles functionalize with polyphenol mixtures derived from pomegranate peel and coffee pulp extract, induce an antiproliferative effect in cancer cells, primarily through modulation of apoptosis-related molecular mechanism. The results demonstrate that the nanoparticle treatments promote activation of apoptotic pathways, triggering programmed cell death that significantly contributes to tumor growth inhibition. A part of this information, we are currently advancing the research by the evaluation of cytotoxicity through LDH assays and analyzing RNA extracts to quantify expression levels of key cell cycles regulatory genes. This ongoing investigation aims to further elucidate the molecular mechanism that are underlying our observed therapeutic effects. And we look forward to sharing these comprehensive results in forthcoming communications. And for me, that will be all. And thank you for your attention.
Okay, so thank you, Dr. Sandra. So the effect is largely dependent on coffee pulp more than pomegranate peel, right?
Exactly. We see a lot of more effects in both HeLa and A5049 cells with the nanoparticles with coffee pulp extracts. Yes.
I have a question. Do people that grow coffee have a lower incidence of different types of cancer?
That grow coffee.
Farmers. The problem is
Farmers.
in Torreón, Coahuila. This is very interesting research that was developed from observing that cotton growers, people that were involved in the cotton picks, those that picked cotton, would normally chew on cotton grains, and they observed that they had lower fertility rates, and that's how gosipol, the only male anticonceptive or contraceptive chemical strategy, was devised or was discovered. So given that drinking coffee obviously doesn't seem to decrease. Actually, I think coffee is directly related to, or at least when I was studying medicine, to gastric cancer and colon cancer, more than a protective effect. But I'm starting to think whether people that grow coffee also chew on the grains. Here in San Luis, we have this important chocolate factory. It's called Constanzo, which makes coffee grains covered with chocolate. They're absolutely delicious. So that might protect me against cancer if I eat several of them, perhaps if I drain the chocolate off. But I'm stuck thinking whether people that are involved in in cultivating coffee or harvesting coffee, if they have a lower incidence of malignant diseases, that would suggest that exposure to coffee husk, or perhaps if they chew on coffee beans like I do, perhaps if they're involved in any other processing of coffee, they would be having this antiproliferative effect of raw polyphenols present in the coffee husk rather than the coffee drink. But we don't know anything about the incidence of cancer among these people, right?
I don't have that information, but it will be interesting. Maybe I'm thinking in skin cancer, maybe because or they are in constantly contact with the husk of the coffee cherry.
But you used, I mean, HEC is a human endothelial kidney cell line, and HeLa is a
HeLa is
cervical
cervical.
epithelium, I think.
Yes,
So
we use HeLa, that is like a cervical cancer cell
yeah,
line.
epithelial cell line. And
A
MDA-MB, what was it? 8549 is lung cancer. Okay.
cancer,
Breast cancer.
cancer.
So skin cancer is not there.
No,
So
it's not. It's not here. MDA-MB, we don't see like a lot of results because it's a very aggressive cell line. In here that we see a lot of good results, and also in lung cancer. Obviously, aligned with the gold nanoparticle that have for themselves like a good anti-cancer activity. The gold nanoparticle.
okay, that's interesting. I've got a last question. When you gave us the incidence of cancer at the very start of your presentation, you mentioned that Asians had the highest incidence of cancer. That seems counterintuitive. I would have thought that they had the lowest incidence of cancer. Perhaps Americans would, or Europeans would have the highest incidence of cancer. But why are Asians so affected by cancers or neoplastic diseases?
Well,
Again,
I don't know.
48%.
They are in like the percentage, and I will think is like for the proportion of population in regions of Asia.
Okay, so it's more people, so you have a higher incidence. It's not corrected for population then.
Yes, it is not like a 48.4 percentage of Asians. No, it's like for the world. So because
Of
there are
all the
like
cancer cases in the world, 48 are in
the world.
Asia because it's a lot more people and there's a lot more
Yes,
cases being detected.
yes,
Yeah,
I
that
think.
would explain why Australia has such a low incidence. Okay,
Yes.
cool. I don't know if anyone else has questions.
I want to ask
Go
a
ahead. Go
few
ahead.
questions. What are the costs of producing this substance?
The gold nanoparticles.
Yes.
They are quite expensive, I have to tell, because the gold salt that we use is chloroauric acid, so it's expensive. But in the part of the polyphenols, well, the polyphenols are super, super cheap.
Okay, and maybe there is an advantage of using this type of nanotechnology, because when we consume all this food with high polyphenol content, they not go to make their effect because of degradation and all that. And when you use that type of technique like nanotechnology, they can target the cancer or the tumor. So maybe it's a way of finding some other techniques that are not so expensive.
Yes, yes, of course. And that was the problem with the polyphenols, because if we, well, it's obviously very healthy to eat a lot of fruits and vegetables, but the polyphenols had to survive all the digestion process in the stomach, acids, and then the absorption in the intestines. So little of the polyphenols can reach the the target organ or the target tumor. We select cervical cancer cell lines and pulmon cancer lung cancer cell lines, because the treatment could be not through digestion, so we can do it like more
Directly.
directly. Exactly.
Okay. Maybe an animal model.
Yes, I think that's the next part of these experiments. Move to a murine
Model.
model. Yes.
Great. Very interesting. Thank you.
Thank you, doctor.
Dr. Teniente, I have another question. In your comparisons, apoptosis and necrosis graphs, you never included a group having only gold nanoparticles without any of the extracts, did you, or did you not?
Yes, we propose to do like a control with gold nanoparticles alone, but to synthesize the gold nanoparticles without using the polyphenols, we have to use other reagents, principally sodium borohydride and hydrazine. So it will not be like the same particle. So that's why we don't use it as a control, because it will not be similar. It will be very, very different.
And can't you use the same process and use a different substrate other than polyphenols, or, I mean, something similar that could serve as a control group?
Yes, we propose to use citric acid. That is usually, bueno, is used for the gold nanoparticles synthesis, so we can do like similar nanoparticles and compare it.
Okay. Well, it's a shame, that technical limitation. So we hope you have success with the next review of your article. We hope you get it approved. I don't know if anyone else has a question. Okay, thanks everyone for being here. Thank you, Dr. Sandra Teniente. You're the first one to present us the webinars in English. We really appreciate it. We trust the following speakers will have similar talent. I hope to see you guys next month. Okay, I'll be contacting you. I'm not sure who's next, but I shall be contacting, and you shall be notified through either WhatsApp or through our Facebook, Genomica Wasl. Thank you very much for your time. Thank you for allowing us to postpone your speak for this week. It's a pleasure to have met you. Gabby, thank you very much for making this possible. See you.
Gracias,
I'm
doctora.
gonna-
Gracias, doctor Cristian.
Thank you. I'm gonna stop recording
Bye.
now.
Nos vemos. Adiós.
Bye-bye.